This is the fourth lecture in a series of introductory immunology lectures. This lecture is focused on B lymphocytes and the humoral immune response. In this slide, you can see a picture, an X-ray crystallographic structure picture of an antibody molecule, and you can see that an antibody binding to an antigen, in this case the influenza virus. And the antibody molecule is comprised of a heterodimer, a light chain and a heavy chain, that interacts with a specific epitope or region on the influenza virus. And this lecture is going to focus on how these antibody molecules are made and secreted during an immune response and their role in mediating immunity. So the learning objectives for this lecture are to describe the role of T cells in promoting B cell activation and antibody production, to describe what occurs in the germinal center, including affinity maturation and isotype switching, and how these events are regulated and why they are important in humoral immune responses. And finally, to describe the different effector mechanisms of antibody and correlate the antibody isotype with the effector mechanism. And so in most cases, in order for B cells to become activated and to secrete antibody, they need T cell help. And so this is just a picture of a T cell and a B cell interacting with each other. And the way T cells provide help to B cells is because T cells have a T cell antigen receptor that recognizes MHC peptide complexes presented by the B cell. So in this case, the B cell is the antigen presenting cell that's presenting antigen to the T cell. And in turn, as a consequence of that activating signal, the T cell provides help and activates the B cell to proliferate and to produce a secreted form of its antigen receptor, the B cell receptor. And so one of the things that's puzzling about how this works is that if you look at lymph nodes or other secondary lymphoid tissue, B cells and T cells are largely spatially segregated. So B cells in brown here on the top panel are in the follicles of lymphoid organs, whereas T cells are in the subcortical region kind of around here. But what happens during an immune response, if you jump over here to day 2 of the immune response, the T cells are labeled in blue. And what you can see that over the course of an immune response, some of these T cells are migrating from the T cell zone into the B cell area, where in turn they can interact with B cells. Then if you jump down to this panel, you can see that in this case it's the B cells that are labeled in blue, the antigen-specific B cells. And what you can see is that shortly after the T cells get to the follicle, you get a large expansion in the number of antigen-specific B cells present in the follicle, and this is a result of T-B cell interaction. And so just to go in a little bit more detail about what T cell help is and the steps involved in getting T cell help for the B cell. And so in this case, the B cell, again, is an antigen-presenting cell. And the way B cells often take up antigen is because the B cell receptor specifically interacts with antigen. And one consequence of that interaction is for that antigenic complex to be internalized inside the B cell, processed into peptides, and those peptides are then represented in the form of an MHC peptide complex. And so if you have a T cell that has a T cell receptor specific for that MHC peptide complex, the T cell will become activated. And so one of the important consequences of T cell activation in this case is for the T cell to express an important co-stimulatory molecule called CD40 ligand. And its expression of CD40 ligand by the T cell allows it to interact with the CD40 molecule on the surface of the B cell. And this provides a second co-stimulatory signal for the B cell. So the B cell gets one signal through the B cell antigen receptor up here, and then a second signal through the CD40 co-stimulatory molecule down here. And these two signals together are enough to activate the B cell and allow it to proliferate. And we'll see what happens during that proliferation reaction in a few minutes. But the other thing that's really important for T cell help is that not only does the T cell express CD40 ligand, but it also produces cytokines. And some of these cytokines will act on cytokine receptors expressed by the B cell. And it's really the combination of all three of these signals. So signals to the B cell receptor, signals via CD40, and signals via cytokines that regulate B cell activation and determine what it's going to do. And the B cell, in this case, typically will undergo several different reactions. that we're going to talk about in more detail in a few minutes. But just to give you a preview of the nomenclature that we're going to use, so one thing that's going to happen is the B cells are undergo what's called isotype switching and change the immunoglobulin type that they're expressing. They're going to undergo something called somatic hypermutation that affects the affinity of the antigen receptor for the antigen. And all of these things happen in a proliferation reaction that occurs in the germinal center that forms in the B cell follicle. And the final thing that's important for long-lived B cell function is that some of these B cells, just like some of the T cells that we talked about in the previous lecture, will become long-lived memory cells and are expanded in number and better able to respond secondarily upon re-challenge with that antigen. And so this process by which T cells provide help to the B cells is often described as linked recognition. And the reason it's called linked recognition is that T cell help to B cells is usually linked to recognition of usually different epitopes on the same antigenic complex. And let's go through what that means in a little bit more detail. So here you have a B cell receptor that's engaging with a specific epitope, in this case on a viral particle. So it's engaging with some protein that's on the surface of the virus. And that results in an activation signal for the B cell, but it also allows the B cell to internalize that viral particle and degrade proteins associated with that viral particle into peptides. Some of those peptides are loaded onto MHC class II molecules and then presented to T cells. And if these T cells have a receptor that's specific for this MHC peptide complex, it can provide help to the B cell. And one of the consequences of that help is to activate the B cell and allow the B cell to secrete antibodies. And the antibodies the B cell are going to secrete are actually a soluble form or secreted form of the original B cell antigen receptor that recognized the viral epitope. So what this means is that B cell activation and antibody production are actually linked to the B cell's ability to recognize that antigen in the first place, and is linked to the T cell being able to provide help to the B cell for epitopes that the B cell internalized. And so this is a complicated thing, and I just want to go through it one more time in a slightly other context, because this linked recognition principle is actually used to make more effective vaccines, as we'll see in the vaccine lecture in the next sequence. So here again you have a B cell that has a B cell antigen receptor, or a membrane-bound form of the antibody molecule. And it's interacting with an epitope here that's associated with a conjugate vaccine. In this case, the vaccine is a conjugate of a bacterial polysaccharide epitope linked. This is the tetanus toxoid protein, and the B cell receptor in this case is specific for the bacterial polysaccharide epitope. And what's happening here is that the B cell receptor recognizes the polysaccharide epitope, internalizes the complex, both the polysaccharide epitope and the carrier protein, this tetanus toxoid protein, breaks up the protein into peptides. Some of those peptides are presented on MHC class II molecules. T cells specific for the tetanus toxoid protein will provide help to the B cell, but a consequence of that help is for the B cell to secrete antibody, and the antibody is specific for the bacterial polysaccharide epitope. And the reason this is important is that, remember, T cell receptors typically only recognize peptides that are derived from proteins. They're very, very, very bad at recognizing other types of antigens like polysaccharides that are commonly expressed on bacterial cell walls. And so it would be hard to get T cell help with a T cell receptor specific for the polysaccharide, but you can recruit T cells to help this B cell by hooking onto the polysaccharide this tetanus toxoid protein carrier. So it's a way of linking T cell help to provide help to the B cell to make antibody specific for the polysaccharide capsular epitope. And so if that happens, then the B cell will secrete antibodies specific for the bacterial cell wall epitope, and those antibodies will neutralize or prevent infection from that bacteria. Okay, so let's go through in a little bit more detail some of the steps that are associated with most cases of B cell activation. So one thing that's important, just like for T cells, is clonal expansion. proliferation of B cells specific for a given antigen. And that occurs in the follicle of secondary lymphoid organs. And when you have a lot of clonal expansion in a follicle, that area of proliferation is referred to as the germinal center. And so really what a germinal center is just a region within the follicle where you have lots and lots of B cell proliferation. And when that's happening, there are several other events that are important for mediating this B cell humoral immune response. As I said before, one of these is somatic hypermutation, and another one is immunoglobulin isotype switching. And the final things are the production of some of these cells to become long-lived memory B cells, and some of these cells will differentiate further into plasma cells. And what plasma cells are differentiated B cells that are really dedicated to secreting lots and lots of antibody at a high rate. And so this is just a picture of the germinal center. And so here's a picture of a lymph node, and this is the B cell zone, or the follicle. And when B cells get activated, they undergo this proliferation reaction where you get the formation of a germinal center, which is indicated as this little yellow dot. That's at different stages of development in this B cell, in this lymph node. So here's a blown up picture of the little yellow dot, or the germinal center. And for this purpose, there's really just a couple things I want you to notice. So one is that within the germinal center is an active area of lots and lots of B cell proliferation. The other thing that you'll notice is that there are special cells within the follicle and now within the germinal center that are designed to serve as a way of aggregating antigen and maintaining the stability of antigen within that site. And these special cells are called follicular dendritic cells. And the other thing that happens is that if T cells have been activated, they will migrate to this follicle and help potentiate the development of this germinal center. And so these things all together, the source of antigen provided by the follicular dendritic cells and the source of T cell help, are required to allow the germinal center formation and to promote B cell activation. And so this is just another slide showing you the same thing. You have lots within the germinal center, you have lots and lots of B cell proliferation. And one of the things that happens is that during these rounds of B cell proliferation, the B cells will repetitively encounter antigen that's on the surface of these follicular dendritic cells. And so the B cells will be proliferating, and they'll repetitively get stimulated by antigen, and also potentially repetitively get T cell help, which are the cells with the red circle as opposed to most of the cells that have the blue circle, which are the B cells. And so depending on the signals that the B cell receives in the germinal center, receiving either through its B cell antigen receptor when it interacts with antigen presented by the follicular dendritic cells, or it receives by physically interacting with the T cell, these B cells will either proliferate and become long-lived memory cells and undergo changes in their antibody molecule that we'll talk about in a few minutes. These changes are referred to as affinity maturation and isotype switching. But the other thing that happens is, just like in most immune responses, whenever you have lots and lots of proliferation, you also have lots and lots of cell death. And so if these cells don't receive the proper stimulatory signals, they will die by apoptosis, or programmed cell death. So this is just a cartoon to illustrate the two main or three main changes that occur to the immunoglobulin molecule during activation in the germinal center. So this is the antigen binding portion of the antibody molecule. And one of the things that happens is the somatic hypermutation process that creates mutations within the antigen binding region of the B cell antigen receptor. And if those mutations result in the B cell antigen receptor having a higher affinity for antigen, these B cells will have a survival advantage because they'll be able to engage with more antigen more efficiently and get better survival signals. And so what happens over time is that when you have more somatic hypermutation that results in a higher affinity of the antigen receptor for antigen, overall you'll get a higher affinity of the antibody response during an immune response. The other thing that happens is that the constant region of the antibody molecule, this part down here, will undergo something called isotype switching. So most B cells, before they're activated, express surface immunoglobulin, and they express surface immunoglobulin of the IgM isotype. And what isotype switching refers to, as you'll see in a few minutes, it refers to switching out the genes that encode for this portion of the molecule, and they'll switch from the IgM isotype to other immunoglobulin isotypes, for example, IgG or IgA or IgE. And then the final thing that happens is, that's outlined in this middle panel, is that if these B cells differentiate further and become plasma cells, instead of expressing the B cell antigen receptor that's a membrane-bound form, they'll switch this portion of the immunoglobulin molecule and be able to, instead of expressing immunoglobulin on the cell surface, they'll secrete a secreted form of the antibody of the immunoglobulin molecule, and this is what's often referred to as antibody. And so you have these three different changes in the immunoglobulin molecule. You have a change in the antigen binding portion, you have a change in the constant region that affects the function of the antibody molecule, and finally you might have a change in the transmembrane portion that facilitates immunoglobulin secretion. And so just to drum this in again, here's a cartoon structural representation of the immunoglobulin molecule. This is the variable portion at the immunoterminus, and this is the portion that engages with antigen. And so this is the part that undergoes affinity maturation that affects antigen binding. This is the constant region of the molecule, and this is the portion of the molecule that undergoes isotype switching. That refers to changes in this region, and it's just changes in the region of the heavy chain of the molecule that alters effector activity of the immunoglobulin molecule, as you'll see in a few minutes. Okay, so just to review the things that are happening during the germinal center reaction, when a B cell gets activated in the follicle, it may proliferate. Those B cells will have somatic hypermutation that results in mutation of the B cell antigen receptor. If that mutation results in a higher affinity of the B cell receptor for antigen, these B cells will be selected to survive. They'll engage with more antigen and get more T cell help, and you'll have more sustained proliferation and maturation, and these B cells will be destined to developing into memory B cells or antibody secreting cells known as plasma cells. However, if the B cell antigen receptor mutates so that the B cell receptor has a lower affinity for antigen, these B cells will not get survival signals. They will not be able to compete with these B cells. The receptor won't be signaled. They won't get T cell help. And it is these cells that will die by apoptosis. And so over the course of an immune response, you'll have selection of cells that have a higher affinity for antigen and to potentiate the development of both memory B cells and plasma cells. And so this is just a picture showing you what plasma cells look like morphologically. So this would be a resting B cell that's mostly all nucleus with relatively little cytoplasm. And when the B cell differentiates into a plasma cell, it looks morphologically like this. The nucleus is condensed and usually is eccentrically placed. And one of the things you can see is that there's a lot of cytoplasm, and the area around the nucleus you can see is a little bit clear. And what that represents is a very well-developed Golgi apparatus indicative of the function of these plasma cells, because what they're doing is they're producing lots and lots of protein, lots and lots of antibody molecules that are secreted out into the extracellular space. And so what these plasma cells do is that they leave the lymph node and they go to extracellular sites. Many of them actually migrate to the bone marrow, where they can be long-lived cells and be a continuous source of immunoglobulin production. And most of the serum immunoglobulin that we all have circulating is a product of these plasma cells that are long-lived and often live in the bone marrow. Okay, so let's talk now a little bit more about isotype switching. So remember, immunoglobulin isotype switching involves changes in the DNA that encodes for the constant region of the heavy chain gene. And immunoglobulin isotype switching allows B cells over the course of an immune response to express different immunoglobulin isotypes. And so this is just a cartoon of the 5 major immunoglobulin isotypes. So you have that are secreted. IgG, IgE, IgM, and IgA. Now, IgD is not secreted and usually is only expressed as a membrane-bound form of the immunoglobulin molecule. But IgG, IgE, IgM, and IgA are all secreted. The other thing you notice is that for IgA and IgM, they're secreted in a multimeric form. So IgM, when it's secreted, is secreted in the form of a pentamer, so 5 antibody molecules hooked together, whereas about half of the IgA that's secreted is secreted in the form of a dimer, so 2 IgA molecules secreted together. And so isotype switching is something that occurs relatively late in the primary immune response, or relatively late after the first time one is exposed to antigen. And so what this graph is showing you is on the x-axis is time, and on the y-axis is the relative antibody concentration. And what you can see in the green humps here is the level of IgM production that's produced after primary activation. You can see it's relatively slow in coming and relatively limited in amount and decays fairly quickly. But one of the things you also notice is that at later time points after this primary immunization, you start to switch from IgM being produced to IgG being produced. But the really dramatic thing that happens is that the second time you're exposed to that same antigen, you may get a reinduction of more primary B cells that haven't been stimulated before, and so you get more IgM production, but you get a lot more IgG production because you have re-stimulation of those B cells that have undergone isotype switching. And so there's a characteristic pattern of immunoglobulins that are produced over the course of an immune response. IgM is secreted first, and then typically IgG is secreted thereafter, and followed by IgA and IgE. And these immunoglobulin isotypes have different immune effector functions, and they're summarized on this slide. So IgM is, when it's secreted, is largely good at neutralizing antigens, meaning that you neutralize the function of those antigens. It prevents viral binding into cells, it prevents bacterial binding into cells, it may prevent the toxins from interacting with toxin receptors on cells, and we'll talk more about all of those functions in a few minutes. And the other thing that IgM is especially good at is actually activating the complement cascade that we talked about in the innate immunity lecture. IgG is usually the most abundant immunoglobulin isotype. It also has some of the same functions that IgM does, meaning neutralization and to a certain extent complement activation, and some other functions that we'll talk about in a few minutes that involve Fc receptor binding. IgA is an immunoglobulin isotype that's especially prevalent at mucosal surfaces, and as you'll see in a minute, that's because IgA is recognized and secreted by epithelial cells that line barrier surfaces, and is transported across those epithelial cells into the gut or other glandular spaces that have exposure to the outside world. IgE is an immunoglobulin isotype that's produced in extraordinarily low quantities, but even though there's very little of it present, it has a very powerful immunological effect because it binds with high affinity primarily to mast cells. And as you'll see in a few minutes, when IgE is cross-linked on the surface of mast cells, that mediates mast cell degranulation and is critical for an allergic inflammatory response. IgD is an immunoglobulin isotype that's not secreted and is present constitutively on the surface of all mature B cell antigen receptors that have not been stimulated with antigen before. So on most naive B cells that haven't been stimulated with antigen before, they co-express surface immunoglobulin in the form of both IgM and IgD simultaneously. So this is just a cartoon to illustrate how IgA gets across mucosal surfaces. So here you have a plasma cell that's secreting IgA, and you can see it's IgA because it's this dimeric form where you have 2 antibody molecules hooked together. And what happens is that there are specialized receptors on the outside portion of these epithelial cells that recognize the IgA molecule and actually transport it right through the cell out to the lumen of the, in this case, of the gut, and is secreted into the mucus in the gut extracellular space. And IgA is important, we think, because it can then interact with microorganisms that are in the gut, for example, bacteria, and prevent those microorganisms from gaining access to the gut epithelial cells and inside the organism. And so to summarize the main antibody effector functions, you can think of them really as three broad categories. So one is neutralization, which just refers to the ability of antibodies to prevent particles, in this case a bacteria, but it could also be a virus, it could be a toxin, from engaging with cells. The other thing the antibodies do, which is kind of almost the opposite of neutralization, it may facilitate the recognition of particles and allow those cells to be phagocytosed by phagocytic cells like macrophages and neutrophils, which is important for the ability of those cells to destroy, in this case, the bacterial cell. And the final thing that, again, we talked about in the innate immune cell lecture is that one of the ways you can activate the complement pathway is that when an antibody engages with antigen, that can activate a cascade of chemical reactions that will result in the formation or an assembly of a series of complement molecules known as the membrane attack complex that pokes holes in the surface of the microorganism where the antibody is bound. And in addition, we also learned that complement can have chemoattractant properties where it can recruit other cells, other innate immune cells like neutrophils, like macrophages, to the site of complement activation. And so this is just a cartoon illustrating the neutralization function of antibody, and in this case, it's the ability of antibodies to neutralize toxins that may be produced by microorganisms like some bacteria. So typically what happens is that the way toxins work is that they bind to specific cellular receptors, they get internalized, and they mediate toxic effects of the cell. But if you have an antibody response that's specific for the toxin molecule, it will prevent the toxin molecule from engaging with the toxin molecule's receptors on the cell, and so it will prevent the toxic effects of the toxin. And that's really the basis by which vaccines to various toxins like the tetanus toxin or the diphtheria toxin work. You're inducing an antibody response to the toxin and preventing that toxin from engaging with cellular receptors and preventing the toxin from causing cell damage. The other way of looking at neutralization is that it prevents infection in the first place. In this case, as you know, viruses can engage with receptors on cells, and that allows the viruses to infect the cell and replicate within the cytoplasm of the cell. But if you have an antibody response that engages with the virus, it will prevent the virus from infecting the cell in the first place. And this is really the principle of how influenza vaccines work. So if you make an antibody response specific for an influenza strain, the antibodies will bind with the influenza virus and prevent that virus from infecting cells in the first place. And the same thing is true with antibodies to bacteria as well. It can prevent infection. And the other thing that's a little bit kind of contradictory from the neutralization aspect of antibodies is that sometimes when antibodies kind of coat a particle, in this case a bacterial cell, the Fc portion or constant portion of the antibody molecule will engage with specific receptors on cells. And in this case, it's a macrophage that's expressing an Fc receptor that engages with the stem part of the antibody molecule or the constant portion of the antibody molecule. And what that does is two things. So one is that it activates the macrophage and also facilitates the ability of the macrophage to internalize the bacteria. So the combination of internalization plus activation allows the macrophage to kill the bacteria that it's phagocytosed. Another important role of Fc receptors is that Fc receptors are specific for different immunoglobulin isotypes. And so this is an Fc receptor that's expressed on mast cells that's specific for the IgE isotype. And what happens in this case is you have Fc receptors for the IgE molecule expressed on mast cells, and any IgE that's produced will bind to these receptors. And so you have all the mast cells that are in your tissue are usually coated with this IgE antibody. And then if the IgE antibody engages with an antigen, as illustrated in this cartoon, the antigen will cross-link the antibody molecules, resulting in cross-linking of the Fc receptors. And what that does is it activates the mast cell to release its intracellular granular contents. And so this is an electron micrograph of a resting mast cell that's not activated, and you can see that it's just full of lysosomal granules that contain histamine and many other inflammatory mediators. But when IgE is cross-linked on the surface of the mast cell, that results in degranulation or expulsion of these lysosomal granule contents into the extracellular space, and that's really the important catalytic event that induces an allergic inflammatory response. And the final thing I just wanted to mention that's related to what antibodies do is they activate complement. And I mentioned complement before in the innate immunity lecture, but just to review, complement is a system of plasma proteins that are activated and mediate pathogen destruction. They're activated by three main pathways, and depending on the pathway, they may be considered part of either the innate or the adaptive immune response. So the classical pathway of complement activation depends on antibody interacting with antigen, which in turn can activate complement. But the lectin pathway and the alternative pathway are independent of antibody, so they're antibody independent and would be more fit better with an innate immune function. But regardless of the way you activate complement, the end result is the same. You activate the effector functions of complement, which really can be summarized here: recruitment of inflammatory cells, opsonization of pathogens, and killing of pathogens. And just to summarize all the aspects of the humoral immune effector response, it's really simple to just think about what antibodies do in this way. They can neutralize, they can opsonize, or they can activate complement. And so you have to remember that neutralization can mean different things. It can neutralize bacterial infection, it can neutralize viral infection, it can neutralize toxic effects of toxins on cells. Opsonization and engagement with Fc receptors can mean several different things. It can mean promoting the internalization of particles by macrophages, as we talked about, but also it can result in the activation of other cells like mast cells and eosinophils that also have Fc receptors that result in degranulation of those cells and mediating the inflammatory responses that are associated with mast cell and eosinophil activation. And finally, complement activation also results in the opsonization of bacteria and facilitates their clearance that way. It facilitates microorganisms' clearance by activating the membrane attack complex and causing osmotic lysis. And then the final thing it does is the complement fragments also serve as chemoattractant molecules and recruit macrophages and neutrophils to the site where complement is being activated.